Thank you, Drew, and thank you for the opportunity to speak with you today, and especially how we're seeing the advancement of our AdenoVerse platform across some new approaches for cell and gene therapy. GenVec is a publicly traded company. I will be making some forward-looking statements, especially in regard to the use of the technology platform. Sorry. We're a pioneering biotech company. We are focused on AdenoVerse gene delivery platform. We have deep expertise in the use of Adno Vectors and established and proven capabilities of moving from scientific concepts to clinical products. We have a clinical stage product for hearing loss and balance disorders that's partnered with Novartis. And we're working with TheraBiologics on a genetically modified neural stem cell program for oncology. And we are expanding the use of our technology in other gene and cell therapy pipeline today, and that's really what I want to talk to you about. So Adno Vectors have a variety of advantages and some very public known disadvantages. Some of the advantages are efficient transduction and dividing cells and non-dividing cells, and the vector genome does not integrate into cellular chromatin, which allows you to not have potential cellular disturbances. The AdenoVerse strengths overcome a lot of the disadvantages to first-generation Adno technology. In particular, we have vectors within this platform with no or very low seroprevalence. We're able to tune the tropism through a variety of means to allow cell-type-specific gene delivery. We have a large carrying capacity for payload, and you're able to get multiple expression cassettes within the Adeno vector genome, if you'd like to express multiple different things from the same construct. We have improved safety using this multiple deletion platform that I will be talking to you a little bit about a little more further into the presentation. And the technology has been administered in over 3,000 clinical subjects. This actually tells you that the GenVec has the expertise to be able to move into the clinic, but also from a regulatory point of view, the technology is well established. It's a commercializable platform with efficient manufacturing processes and attractive cost of goods moving forward. And we have a strong IP position across this new technology platform. How we see the use of the platform is across a variety of new ways. From regenerative medicine, which is at the core of the hearing loss product, where we regenerate sensory cells in the inner ear, through vaccines, which is molecular vaccines using Adno, is one of the hallmarks of the technology, and you can still do that with this new technology, to some of the new approaches that we're looking at, and I will spend a little time with today on cell therapy, and then thoughts as you move forward with some of the new technology approaches for gene editing and a few other things. How do we do this? Is that the AdenoVerse platform is actually a library of different vector types. And this is multiples. There are over 27 different types of adenos that we built vectors out of, and from that, modifications thereof that encompass about 80 different things today that you can do with different vectors that allow us to think about distinct sets of vectors that can either be used for sort of the standard approaches for the way people think of adeno for eliciting an adaptive immune response, or for some of the new approaches that we see the use of this Adno platform for more immunologically stealth delivery. And in particular there, what is a core component of what we're doing is the program in the inner ear, which is for otology, that really required the ability to get into these cells without a lot of perturbance, and we can't do that with this platform. There are known adeno vector limitations, so I bring those up here. One of those is pre-existing immunity. All of you have had an Ad5 virus infection, and so you have antibodies, against that vector type, and that is the classic vector that people have used in the field. We have within AdenoVerse, vectors that use proprietary backbones from rare human types of adenos and non-human primate adenos that have low or no seroprevalence in the human population to overcome this limitation. There have been noticed innate immunity-associated toxicities with first-generation adenos in the past, and GenVec's vectors can be deleted of all of the early regions that matter from E1, E3, and E4 that limit the expression of viral proteins and other cell modulators that are responsible for this innate immune response. The other aspect to adeno, when you deliver it systemically, is that the majority of the Ad5 dose goes to the liver. There's vector liver sequestration. And within this platform, there are vectors that appear to not be sequestered into the liver and could be suitable for systemic delivery. So what we see with this platform is the next generation of adeno vectors with unprecedented potential, and we're looking at expanding those. So I do want to spend a little bit of time on this slide, and I know that I'm probably the only molecular virologist in the room, so that's the bottom schematic. But the top parts of this are real important for you to grasp. I mean, we can delete and manufacture vectors that are deleted of E1, what's called E1, E3, and E4. And within all of those regions are viral proteins that either transactivate other viral genes that you don't want to have happen, or they modify the host cell in some way. And so by deleting all of these, you basically build a vector that can get into those cells with a lot less perturbance. You also then, by the fact that you've deleted a large part of the adenovirus genome, provided a larger packaging capability. And because of all of these other genes that are deleted, not only do you lessen this innate immune response to the adenogenome, but also all of the cellular process perturbance that can occur. So I do want to spend one slide on this regenerative medicine product I brought up that is partnered with Novartis to treat hearing loss and balance disorders. The vectorology for this product concept actually utilizes this multi-deleted Ad5-based vector. It's very important for this because you want to be able to get into those cells without any further perturbance, because we want to get to what are called supporting cells in the cochlear space and the vestibular space to transdifferentiate those by expressing atonal into sensory cells. When you lose your sensory cells in your hearing space or in your balance space, you don't grow those back. And that's one of the largest forms of hearing loss. And this drug is set up to actually regenerate those sensory cells and provide the means in which that you can reestablish your hearing. We've shown that we can deliver the material to the cochlear and vestibular system. We've demonstrated efficacy and safety in a variety of models, both in vivo animal models and ex vivo. And this is currently in an ongoing phase 1/2 proof of concept study that's designed to evaluate both safety and efficacy. We're also working with Karen Abudi at City of Hope and TheraBiologics on modifying their neural stem cell technology for directed enzyme product therapy. Basically, the adeno vector that we're using expresses carboxylesterase, and the NSCs are transduced and then delivered to patients. The first clinical target is in glioma, and looking at other aspects to this. The cool thing about NSCs is that they penetrate the tumor microenvironment, and in this particular setting, the secreted carboxylesterase can then provide a radius of action throughout the tumor. Carboxylesterase converts irinotecan to SN38, and that's 1,000 times more potent toxic to the tumor cells. And this second generation molecule is expected into the clinic end of this year. So that was our first foray into cell therapy. And so we came back and started thinking about, well, could we use the technology in a variety of other ways? And so we started looking at both T and NK cell transduction, and we think that this technology has multiple applications for adoptive cellular immunotherapies. And I do want to walk you through some of this data. This is screening our AdenoVerse for vectors that efficiently transduce primary human T cells. And there are 4 winners depicted here from the AdenoVerse screen that show upwards of about 80% transduction of T cells. Prior to this, if you look at the literature, Adno Vectors are known to poorly transduce T cells down in about a 5% range at best. With some modification, you can get up to about 15 to 25%. So we were very pleased with this. And with a little modification, with one of these vectors, we've actually been able to show that we can get around 95% of those primary human T cells transduced. So we're really excited about looking at opportunities to use these vectors to modify T cells moving forward. We've also started another screen looking at the delivery to NK cells. Same sort of idea here, and this is just an early stage of this, and I show you a few of these winners where the classic Ad5 vector has very limited transduction capability into NK cells, but some of the constructs through the evaluation of this initial screen, we're seeing some phenomenal transduction scores, and we're just at that point of really evaluating this a little further. So we think that the AdenoVerse has a variety of solutions, for cellular immunotherapy requirements, high level of transduction. We can build, high level and sustainable transgene expression. The majority of the manufacturing constructs that you would think about we could do with Adno Vectors today. We think that the technology in itself, could be applied to reduce, on-target off-tumor effects, just because you're not expressing, these protein products that you would want all the time. However, you could, because you could, deliver through, some kind of gene editing approach to, to make a stable, transformed cell as well. This is where we've been, and with some of the targets, for the vectors, the eye, the ear, lung, cardiac, a few other places in the body, and on the right side of this is to depict that these 2 ex vivo applications that we're looking at, both for these neural stem cells as well as these immune system cells now that we're looking through. We also think that the platform has applicability and provides a unique solution for delivering gene editing technology. It has more of a hit and run aspect to help limit off-target effects. We can get a large carrying capacity for your payload, so you can start thinking about, in a number of situations, having most of the pieces and parts that you want to deliver in a single vector. And as I brought up before, low immunogenicity for a variety of these vector types that we're moving forward. GenVec's approach is a collaborative one. We see that the AdenoVerse platform offers multiple opportunities for product development and licensing. It's very, very much supported by our extensive translational expertise, and we are actively exploring opportunities to expand the application of the platform and to add complementary assets or technologies to our current portfolio. And we really are looking for that open, innovative approach to collaboration, and would like to be able to talk with you more to expand the reach and really unlock the potential of this new vector technology, and to really think about how this can be used for new medical treatments. I'm here at the meeting, and Sebastian Melavest is as well, and this is our contact information. If you'd like to talk to us, please don't hesitate, and we are very open to what your thoughts can be to help develop this technology platform further. Thank you very much.